Welcome to the lecture discussing the nature of antigens and the major histocompatibility complex. We're going to learn a lot of new terms in this lecture. We are going to discuss the factors that influence the immune response, traits of immunogens, so we're going to define that as well. We're going to define epitopes, haptens, and adjuvants, and the relationship of the antigen to the host. And we're also going to discuss and explain or describe the major histocompatibility complex and its role in the immune response. There are several factors that influence the immune response in every person. So, have you ever been so stressed out that you got sick? So that is a legitimate reason that can decrease your immune response. The age of a person affects the immune response. Older individuals and very young neonates, they have a different immune response because older individuals, they have a decreased response to antigens. And we are even going to learn in the blood bank that some of their antigens may not even show up in our laboratory testing. And for neonates or newborns, their immune system is not fully developed yet, so they have a diminished immune response as well. The overall health of a patient or person influences their immune response, and overall health means their nutrition status, whether they're malnourished or eating healthy, fatigue, stress, and also genetics plays a role in our immune response. The route of inoculation. So there are several routes that we can come in contact with antigens. It can be through intravenous inoculation, where that means that it's injected right into our veins. Intradermal is between the layers of skin. Subcutaneous is beneath the layers of skin. And oral administration is by mouth. So the route of inoculation, that's mostly talking about vaccinations, or in particular, maybe the tuberculosis skin test. That would be a subcutaneous or intradermal test. The dose of the antigen is going to influence the immune response. Usually, the larger the amount of the immunogen exposed, the greater the immune response. Although there is one caveat: if the dose of the antigen is so large that it sort of overwhelms the immune system, and so T cells and B cells may become tolerant. It's not very well understood, but it could be that there's so much of the antigen in the system that the T cells and B cells thinks it belongs there. And also the genetic capacity or predisposition for getting certain illnesses. And we're going to talk about that later in this chapter. It's pretty interesting. Immunogens are macromolecules capable of eliciting an immune response. Immunogens are macromolecules, so large molecules capable of eliciting an adaptive immune response by inducing the formation of antibodies or sensitized T cells. And we're going to talk more about sensitized T cells in this chapter as well, in this lecture. Immunogens can react with antibodies or sensitized T cells, and all immunogens are antigens. And this might be a test question. An antigen reacts with antibodies or sensitized T cells just like immunogens, but they may not create an immune response. Antigens are capable of creating an immune response, but not every antigen may create an immune response. Antigens do stimulate antibody production, but not all antigens are immunogens. The immune response is caused by a combination of three factors. And the three factors that cause an immune response are the nature of the immunogen itself, the genetic coding of the major histocompatibility complex molecules that have to combine with an immunogen before T cells can respond, and we're going to talk about that later. Immunogen processing and presentation cells. those are these three cells that can do this. Macrophages can process and present immunogens, dendritic cells, and B cells. And they have to process these immunogens and present them on the MHC markers that are on every cell to the T cells. And we're going to go into that again. Don't worry, it's a little confusing in the beginning. Okay, so back to the traits of the immunogens. We said that they are very large molecules, so the immunogenicity, or the ability to cause an immune response, depends on the size of the immunogen. Usually the bigger they are, the more potent they are. So these Daltons, that's the size of how proteins are measured. We don't really care about this, but just know that 10,000 would be a smaller one, 100,000 would be a larger protein or macromolecule, and usually the larger the immunogen, the more potent or larger immune response that it causes. Foreignness, the degree of foreignness, so the more different that the immunogen is from the host will cause a higher immune response, more significant. So for the immunogen, it has to be recognized as not belonging there, so as not as self. The best immunogens, and this might be a test question, the best immunogens are very large proteins or polysaccharides. Proteins are actually a little bit better, and these have multiple epitopes with many specificities, and we're going to define epitope in just a second. Pure nucleic acids and lipids are not immunogenic by themselves. They're too small, although a response can be generated when they're attached to a carrier molecule. So just know that nucleic acids like RNA and DNA and lipids are not immunogenic by themselves. They do not cause an immune response. And we talked about genetic predisposition, which is actually different, a little different than actual genetics. So I like to use the example of HIV. So we know that HIV is a virus that likes to attach to our white blood cells, and it has to attach to CD4 T helper cells in particular. But on that CD4 helper cell, there has to be a certain receptor called the CCR5 receptor. So this small protein, this one particular protein. So if people have mutations in their CCR5 receptor on their T cells, or they don't have that receptor at all, they can be immune to HIV, because HIV has to have that receptor to get inside of our T cells. I just think that is incredibly interesting. Okay, here we go. Definitions. Epitope is an antigenic determinant site. So what does that mean? That is the key portion of the immunogen, or antigen that causes immune response, recognized in the immune response. There can be linear epitopes or conformational epitopes. Linear epitopes, pictured here, is just a sequential. They're sequential amino acids, one following after another in a single chain. So that's a linear epitope. So then we have the conformational epitope. So the conformational epitope has a folding in one or more of its chains, so that amino acids from different segments are in close proximity to each other. And that's indicated right here. So if it wasn't folded, these two amino acids would not be close to each other, so the immunogen could not recognize it. So those are the definitions of epitope. Make sure that you learn those, because you will see them again and hear that epitope a lot. Adjuvants. Another term. Adjuvants it's a very small molecular weight thing that's administered with an immunogen, in particular a vaccine. Let's use that as an example. It can make immunizations more effective because adjuvants enhance antibody formation, and it works by targeting antigen-presenting cells. It protects immunogens from degradation. It increases the shelf life, which comes in handy for vaccines. It allows for a longer response time and it attracts more immune cells to the injection site. It's used to accelerate and enhance the immune response and increase the duration of protective immunity. So adjuvants kind of boost the immunizations. So let me give you an example of an adjuvant. An adjuvant would be something like an aluminum salt. That's a preservative in a vaccine. So it would increase the shelf life. It would make the molecule a little larger to increase the immune response. So that's an adjuvant. Haptens, another term to learn, is a low molecular weight molecule with only one binding site. Make sure that you make a note of that. It's not in your guided notes. So haptens, low molecular weight molecule with one binding site. It's a non-immunogenic material that create new antigenic determinants when combined with the carrier. It can react with the antibody even without being complex to a carrier molecule. Now, precipitation and agglutination reactions can occur, and precipitation and agglutination reactions are just two of the test methods that we use later on. So just file this away as in maybe something to pull up later. You don't need to know what precipitation and agglutination means just yet. However, when haptens are bound to carriers, they can help form precipitation and agglutination reactions. And so here's the beginning of precipitation and agglutination. So let's give some examples of haptens like we did for adjuvants. Examples of a hapten situation is a catechol in poison ivy. So catechol is one of the chemicals that makes poison ivy active. So this catechol is not large enough by itself to cause an immune response. However, when you get this poison ivy catechol on your skin, it binds with the tissue proteins in your skin, and that's what causes the immune response. With penicillin or other drugs by themselves may not cause an allergic reaction or immune reaction, but once it forms or joins with a protein, which most drugs need to do to be active in our bodies, then it can cause an allergic response, and it can be even life-threatening. So haptens can be very serious. Now we're going to define antigens as they relate to the host. So autoantigens, those are antigens that belong to us, the host, and normally they don't evoke an immune response, so we don't want to be allergic to ourselves. And if immune response does occur, it's called an autoimmune condition. So you've probably heard of lupus or rheumatoid arthritis. Those are autoimmune diseases where your body's immune system goes awry and starts attacking parts of your own body. And you can have, you can make auto antibodies to your own antigens, like down to the smallest thing, like your own DNA or your own histones, which are the proteins that DNA wraps around. It's so interesting and crazy, and there's a whole chapter just on autoimmune diseases. Alloantigens are antigens from other members of the host species. For example, that's what we work with in the blood bank, because if you donate blood to another human, that person, the donor, the recipient has the potential to develop an alloantibody. So to go with every antigen, there's an antibody to go with it. So alloantigens are from members of other host species. So if I donated blood to you, that would be an alloantigen from person to person or from plant to plant within the same species. Heteroantigens are antigens from other species. For example, if a flu virus goes from an animal to a human, that is a different species. It's a heteroantigen. And so an example of that would be the swine flu or COVID-19. So we know that swine flu jumped from a pig to a human, and COVID-19 is believed to have jumped from a civet cat to humans. Heteroantigen further, like from plant to human or from microorganism to human. So the bacteria causing strep throat that gets into introduced to our body, that is an example of a heteroantigen. Now, heterophile antigens are, see, it's like different but kind of the same. So phile means loving, hetero means different. So these exist in plants or animals, but they are identical or closely look like in structure, so that an antibody from one species will cross-react with the antigen of another species. So, for example, the antigen from the pneumococcal bacteria can be positive with our B blood group antibodies. So those can be similar. So it's a human and bacteria, but they're so closely related that they can react with one another across species. Also, it's thought that naturally occurring anti-A and anti-B antibodies may be due to exposure to heterophile antigens. And that just means that if you are an O type person, you will have automatically, without ever having a blood transfusion, you will have antibodies to A blood type and B blood type circulating in your system already. And for example, if you are a type B blood person, you will have antibodies to A blood in your blood already, and you've never been exposed. So those are examples of heterophile antigens. Okay, now to the major histocompatibility complex molecules. We call these MHC for short. These are linked to the genetic capability that allows the body to mount an immune response. Okay, these have been found on some classes. We're going to talk about class 1, class 2, and class 3. Class 1 MHC molecules have been found on all nucleated cells in the body. They play a very important role in the development of humoral and cellular immunity, and formerly they were known as HLA, human leukocyte antigens, because at first we thought they were only on leukocytes or white blood cells. But after further research and through discovery, we found that they are not only on white blood cells. So now we call them MHC instead of HLA. So why do we even care about these MHC molecules? Because their main function is to bring antigen to the cell surface for recognition by T cells, because T cell activation will only occur when antigen is combined with MHC molecules. Now, isn't that the coolest thing? It's so minute and it sounds so complex and complicated, and this is going on in our body in cells all the time to keep us healthy, and we don't even know it. And it's just so exciting that we know how this works on the molecular level. So I have a picture for you of a T helper cell, and these are MHC molecules inside of a B cell. It's engulfing an antigen, it's digesting them with enzymes, it's breaking them into portions that are digested, and then these MHC molecules carry these digested proteins from inside the cell to the surface of the cell so that then they can be recognized by the T helper cells. Now, isn't that just amazing? To me, that is just such a beautiful model, beautiful system. So when binding occurs with a T cell receptor on a CD4 positive T cell, that's the T helper, and I can remember that CD4 is on T helper because a 4 kind of looks like an H. So the T helper cell recruits and triggers a B cell response, resulting in antibody formation, and that's what we want for a long-term immune response is antibodies. So it's just so cool. But without those antigen presenting cells and the MHC molecules on the outside of our cells to present these antigens to CD4 T helper cells, we can't get antibody formation. So it's very important. So again, why do we care about MHC molecules? Because MHC molecules must combine with an immunogen before T cells are able to respond. And T cells can only see and respond to antigens when they are combined with our MHC molecules. Antigen processing. So this is what happens. Before a substance or immunogen can stimulate an immune response, it must be subject to antigen processing, which involves enzymatic digestion to create small peptides that can be complex to the MHC molecules on the outside of the cell to present to responsive lymphocytes. So again, here's the picture a little bit bigger. The immunogen or antigen combines on the outside of the cell, it's engulfed, it's digested with enzymes and lysosomes, it's broken into very small peptides. The MHC complexes to those peptides and then carries it to the outside of the cell. And there's a really cool video that we're going to watch that sort of uses jelly donuts and as an example of antigen processing. Again, why do we care about MHC molecules? Because these MHC molecules determine whether or not a transplanted tissue is histocompatible or rejected. So these MHC markers are on our cells and they tell our immune system that, hey, these cells belong here. So if a tissue is transplanted and our immune system doesn't recognize those MHC molecules as belonging there, as ourself, it's going to start an immune response and reject that transplanted tissue or organ. So what's also important is the genes that code for MHC molecules. These are found in the humans on the short arm of chromosome 6, and they're divided into 3 categories or classes, class 1, 2, and 3. Class 1, they are designated as A, B, and C. Class 2 is found on the D region, is designated as DR, DQ, and DP. And Class 3 is found in a region between 1 and 2. And just for fun, here's a picture of the short arm of chromosome 6. MHC molecules, classes 1 and 2, are involved in antigen recognition, and they influence the antigens to which T cells respond. Class 3 is secreted proteins, but they are not expressed on cell surfaces. And here's a picture of chromosome 6, the short arm, and the locations of class 1, 2, and 3. Don't memorize that. The MHC genes are very closely linked, and so the inheritance for the grouping is known as a haplotype. And one haplotype is inherited from each parent. And don't memorize this part, but you can see that HLA-A or MHC-A has over 2,000 different alleles. B has over 2,600 and C has over 1,500. So you can see there is exponentially, there is just a huge range of different combinations that can exist. At each of these loci, there's a possibility of multiple alleles or alternate forms that code for slightly different varieties. An individual inherits 2 copies of chromosome 6, so there's a possibility of 2 alleles for each gene on the chromosome, unless that person is homozygous. And this one is not so important, just so you know where we're going with this. The particular MHC molecules produced also determine the responsiveness to individual antigens. So again, this is how genetics play a part in your ability to respond to immunogens. MHC genes are also co-dominant, so that means every gene inherited is expressed. Inherited together is a package called the haplotype. Each haplotype consists of a package of genes for A, B, C, DR, DP, and DQ. The full genotype would consist of 2 of each gene at a particular locus. The polymorphism of the MHC system has implications for organ donation and paternity testing, and I'm fixing to give you an example of what that means. So each person's MHC type is as unique as their fingerprint. That's the point that we're getting to. And this can be used in paternity testing as well. This is what's used in organ donation matches as well, these MHC types. There are 2 types of blood paternity tests, the HLA or MHC test. and it gives us a probability of 95% or greater if the presumed or alleged father is the actual biological father. Now, most in modern times or in today's world, generally the DNA paternity test is better, but I'm using this example to explain the MHC haplotypes. Okay, so I'm going to give you this little problem, and we're going to figure out which one is the father. So there's a child, here's their genotype, here's the mother, here's her genotype, here's the donor 1 and donor 2. So we don't know which one of these two, if either one, is the father. So the first thing we want to do is find which genes from the child match the mother. So there's A6 in the mother and A6 in the child. So A6 comes from mom. B26 in mom, B26 in the child, CW2 in mom, and CW2 in the child. So now we're looking for, does the father have A15? A15, A15. So donor 1 so far, we have a match, partially. B2 in the child, oh, B2 could have come from donor 1. Then CW5, oh, there's no CW5 in donor 1. So donor 1 is not the father. All right, donor 2. Does donor 2 have an A15? Yes. Does donor 2 have B2? Yes. Does donor 1 have C5? Yes. So, congratulations, it's a boy. Donor 2 is the father. So this is how tissue typing is done, as well as paternity testing based on the MHC haplotype. Class 1 MHC molecules are the MHC molecules expressed on all nucleated cells. Class 1, you do need to know these. They differ in level expression, however, based on the cell type. There are the most MHC class 1 molecules on lymphocytes and myeloid cells. They have the lowest or undetectable number on liver cells, neural cells, muscle cells, and sperm. These present peptides made inside the cell to the CD8 T cells. So you do need to associate CD8 cytotoxic T cells having a relationship with class 1 MHC molecules. MHC class 1 molecules are the watchdogs of viral, tumor, and parasitic antigens. Class 2 molecules are found primarily on antigen-presenting cells, such as B lymphocytes, monocytes, macrophages, dendritic cells, and thymic epithelium, so in the thymus, your epithelium. These contribute to antigen recognition by CD4 T helper cells. They alert CD4 cells to foreign protein, for example, bacteria. Class 1, there's peptide interaction. The main role of the class 1 and 2 MHC molecules is in antigen presentation. And again, these have to present the antigens to T cells so T cells can see and respond when they are combined with MHC molecules. Class 2, also peptide interaction. These molecules are synthesized in the endoplasmic reticulum and associated with a protein called we don't even care. But the MHC2 peptide complex is transported to the cell surface. So this is the one that's going to transport the antigens to the cell surface and complex with CD4 T helper cells. So remember that class 1 is associated with CD8 and all nucleated cells, and class 2 is associated with T cells, CD4 T helper cells. All right, let's see where we are. Okay, important, the clinical significance of MHC, like we've said, they're important in tissue transplant procedures. Class 1 and 2 can induce graft rejection, so on those organs that do have a lot of MHC receptors on the cells, they have to be closely matched. The MHC molecules also appear to play a role in the development of autoimmune diseases. Here are some diseases associated with the MHC alleles. Do not memorize this, but just know that rheumatoid arthritis, we talked about that as an autoimmune disease, type 1 diabetes, celiac disease, those are associated with MHC molecules. Here's a nice table that I made for you so that you can quickly see where MHC class 1 and 2 and 3 genes are located, what products, the location, and their function. And then the summary. Immunogens are macromolecules that elicit the formation of immunoglobulins or sensitized T cells in an immunocompetent host. An antigen denotes a substance that does not elicit a host response necessarily, but reacts with antibody once it has been formed. Immunogenicity is influenced by factors such as age, health, route of inoculation, and genetic capacity. An epitope is a determinate site of immunogen recognized by B or T cells, and it can be linear or conformational. Haptens are non-immunogenic substances that must be combined with the carrier to provoke an immune response. Adjuvants are substances that can be mixed with antigens to enhance the immune response. Autoantigens belong to the host. Alloantigens are from the same species as the host, but not identical. Heteroantigens are from other species, and heterophile antigens exist in unrelated species, but their structure is so similar that they cross-react with antigens from a different species. MHC encodes class 1 and 2 molecules, which play a major role in antigen presentation to the T cell. Class 1 and 2 also bind peptides within cells and transport them to the plasma membrane, where they can be seen and recognized by the T cells on the outside of the cell. And class 1 molecules present antigens to CD8 T cells, triggering a cytotoxic reaction, while class 2 molecules present antigen to CD4 T helper cells, which are involved with antibody production. And that concludes the lecture on the nature of the immunogen and the MHC complex.